Back again. So this is video 3 of the cell reproduction chapter, and that's chapter 10. So we finished up last time just talking about sort of the steps of cell reproduction, and particularly with looking at mitosis and how a cell divides into two identical cells or daughter cells when it needs to for a lot of reasons, including it needs to replace itself or the organism is growing, etc., etc. But now we want to look a little bit deeper, and this is to talk about how cells actually decide or regulate whether or not they're going to go through the process of mitosis and make a new cell. So there are a lot of really important features of cells, things which are going to control whether or not a cell does go on and make another copy. So first of all, it should be known that a new cell, so when we create a new cell from an existing cell, that new cell has to be identical. It must duplicate what that original cell is in all respects. So it's really super important that to prevent any mistakes happening, because mistakes will influence what that cell is able to do, its function. So for example, they give the example of mutated chromosomes. That can be a problem. So because of that, because of the importance of having high fidelity in the offspring cells or the daughter cells, what occurs is that we have a number of built-in regulatory mechanisms to stop a cell or to check to see if a cell is ready to move on to mitosis. So there are three main ones. These three checkpoints are found, one of them at the end of G1. So as a cell is sort of getting to the end of its first gap phase, there's a checkpoint that's going to prevent it from going in any further to the next phase, which would be the S phase. And we'll talk about that. There's another checkpoint at the end of at G2 before the cell actually starts to divide. And then there's a checkpoint in the middle of mitosis that is basically a proofreading operation to be sure that the new cells are in good shape. So let's go on. So this is just a kind of a funny little cartoon I found. It talks about, this violation of these checkpoints. So cells will need to fix mistakes, just much like we do when we do work, in order to move on and make a new copy of themselves, or they face a condition called apoptosis. Apoptosis is a programmed cell death. In other words, the cell is not going to move on and make another copy. It is destined to be recycled in a sense, and that's called apoptosis. So the things that are going to make those determinations, there are a number of them. One, there can actually be some chemical messengers that can come in, external triggers, that can initiate or inhibit the cell cycle, such as the death of nearby cells. So you fall outside and you scrape your knee, and a bunch of those cells die and need to get replaced. So that can be an external trigger to tell the healthy cells that are around it, okay, now we need to make new copies and replace these cells that have been damaged or destroyed. Another is the release of growth hormones. So sometimes hormones are released, and that is the trigger to say, all right, we need to make more cells. The other thing that can control cell cycle is crowding. You don't want too many cells. You don't want them starting to stack up on top of each other, so that can also control or inhibit a cell from going on and making too many copies. And there's also some internal factors that are going to regulate whether or not a cell moves on to the next phase. So these checkpoints, as we just mentioned, are found, and let me move this a little bit here, I can move over here so you can still see me a little. These checkpoints are along those areas. So here's G1 in this particular diagram. Here's this green that represents G1. So this is the gap 1 phase. I sometimes think of it as sort of the growth phase. So right here at this red line, it is the checkpoint. And one of the things it has to ensure is that before it moves on, it wants to be sure the cell doesn't want to move into here if there's already some DNA damage, unless that can get repaired. So that's a checkpoint right there. When you go into the S phase here, and you're moving along, that also has a checkpoint that basically the proofreading goes to see whether or not there's any if there's any mistakes done in the replication of the DNA. Then we go into G2, and there's another checkpoint here. So this again is just an additional checkpoint looking at DNA damage. Then we're now into mitosis. So mitosis moves along, and right around this metaphase point where we see those chromosomes lined up in the middle, there's another checkpoint, and that's to be sure that all of these fibers, these spindle fibers that were reaching out and attaching to those centromeres, or the other term was the kinetochores, those little hooks that are right there and holding those two sister chromatids together. We want to be sure they're all lined up and they're all attached, so that when we pull, going one direction or the other, each of those in that sister chromatid pair, each one moves in an opposite direction. So that's another checkpoint. So these are some classic checkpoints that ensure that the cell doesn't move on, doesn't make more copies, unless all of these things are correct and in place. So here's kind of a listing again. At G1, there are external influences that are considered, and to determine whether or not we have, some injury. Do we have enough energy? Do we have enough materials? That's all important at G1. There's also a check to see that DNA hasn't been damaged, and that if a cell isn't going to meet all those requirements, it's not going to go into the S phase. So if that's true, there are a couple of options a cell has. One is just to stop at that point and try to correct a problem. And the other is to enter something called G0, which I mentioned before. It's kind of a holding pattern for a cell, and it allows it to wait there in this holding pattern until conditions may improve, so that it could move on and make a new copy. So here again, there's a lot of different diagrams of cell cycle, and I've pulled some off just to show you some other versions here. But here's where G1 is on this diagram. Okay, so here's that G1 checkpoint. And what you see here at this G1 checkpoint is this right here. These things we're about to talk about, which are cyclin-dependent kinases, which is that CDK, and then cyclins themselves, which are a chemical that is produced that during in response to some signal. So we're going to see that. So let's start with this well, actually we're not starting, we already did G1 a little bit, but we're going to go back to that. The G2 checkpoint is the one that is going to be sure that there's no major problem in the replicating of that new DNA. So this is really important. We want to be sure that we have the right number of chromosomes, that all the chromosomes are correctly made, the copies of those chromosomes. And so at this point, if it is not ready for whatever reason, then there can be kind of a pause, and that will allow, in some cases, for that DNA to get repaired, so that there are ways to repair DNA, and we'll see that hopefully in the next section. So that's the G2 checkpoint. Then we get to that M checkpoint, and so up here, I kind of pulled this diagram here. These are the different phases of mitosis, starting at prophase and eventually going to the division of the cell by cytokinesis. Right around metaphase, that middle one that we talked about, this is where we look to see that everything's well attached, so that each of these new daughter cells that forms has exactly the right number of chromosomes in each cell. So we don't get an extra chromosome or a couple of extra chromosomes or too few chromosomes. So very important. Now, one of the things that the cell does is, in order to regulate this, is it has both positive and negative regulators. Positive regulators are going to help push the cell into the next cycle. Negative regulators are going to ensure that the cell doesn't move on until everything's right. So we're going to take a look at a couple of those. Cyclins are produced, and cyclins are things which will bind with something called a cyclin-dependent kinase, or CDKs. And when they do, so these cyclins that are produced are going to bind with one of these cyclin-dependent kinases and then activate it. So that cyclins are a cell cycle regulator, and we'll look at a couple of specific ones. So here you can see a couple here. So during G1, we have cyclin D that increases in concentration. In G1, we also see another one, so this is cyclin E. Another one that begins in G1, but later in the game is cyclin A. And then here's another one, cyclin B. So these proteins, which are produced in the cell, are going to fluctuate depending on where in the cell cycle we are. So you're going to see an increase at certain times. Notice more of this is being made as we go further along in the G1 phase. It's kind of at its height right here. And not to get too crazy about all this, we'll talk a little bit more about the specific ones. So the increase in cyclin usually is a signal that the cell is getting ready to go to the next step. And how it works is that cyclin binds, so this purple here is cyclin, and then the CDK, or the cyclin-dependent kinase. Remember kinases? We just talked about those. Those are the ones that like to put phosphate groups on things and etc. So, and usually that is an activating mechanism. So here, when we get cyclin binding to CDK, we get a kinase here activity. So we're going to get a phosphate group on it. And then that phosphate group that's on this kinase can phosphorylate, in other words, donate that phosphate to some other protein, which ultimately that target protein then is going to cause the cell cycle to move forward. Now, there's also some negative regulatory mechanisms, and the best one are these ones here, RB, which stands for retinoblastoma protein, P53, and P21. And they act mostly here at this G1 checkpoint, and basically when they are not phosphorylated, they're going to inhibit the cell from moving on into the S phase. But when they become phosphorylated, that disinhibits them, which allows the cell to move forward into the next phase. Now, the details on this I'm going to get into a little bit more in class for these negative and positive regulators. So I'm going to quit there and we'll move on.